Now we're going to take a little bit closer look at the stage where the chromosomes are copied and talk exactly about what's happening there. Okay, so we've zoomed in on that stage. Let me just show you again where that is. So we're going to look at going from here to here. Okay, so we're going to start with this cell up here, and we're going to see what goes on here to make these chromosomes copied. All right, let's try that again. So, starting with that first one, again, we've got one chromosome from the mom in blue, one chromosome from the dad in yellow, and we need to copy those or make a little bit extra DNA, twice the amount of DNA for each one. The little circle in the center is called the centromere again. That's where the pairs actually connect to each other. So we've got a copied chromosome there for the dad. Notice the centromeres are connected. And a copied chromosome there from the mom. Again, the centromeres are connected. So now we've got twice the DNA for each of our chromosomes. And we call this set, this pair of chromosomes that are one from the mom and one from the dad, a homologous pair. The prefix homo means same. Now, why are they considered a homologous pair? What makes them the same? Well, they're the same in shape, they're the same in appearance, they're the same in length, but they're also the same in genes. Okay, so you want to make a note of that to yourself. Homologous pairs have the same length, same shape, but they also have the same genes. Now I'm going to show you what that means here in a minute. If we look at the genes on these pretend chromosomes, we could say they maybe carry the eye color gene. The eye color gene is an area of the DNA that tells your body what color eyes you're going to have. So I'm going to use my parents as an example. Maybe my chromosome from my dad carries an eye color gene, and his eye color was brown. But my eye color from my mom is on a different chromosome. She gave me different chromosomes in her eggs. So in that case, she still has an eye color gene, but her eyes are green. So in my cells, I have one chromosome from my dad with the eye color gene, and one chromosome from my mom with the eye color gene. So they have the same gene on them, the eye color gene. But what's different is my dad's sperm brought a different version of the eye color gene than my mom's eggs. So in me, I have a combination of those two versions. Again, both of these chromosomes have the same gene, but I have different versions of that gene because one of the chromosomes came from my mom and one of the chromosomes came from my dad. So this is a homologous pair. It's a pair of chromosomes, again, the same size, same shape. They have the same genes on them, eye color genes, but they are different versions. In humans, we have 23 homologous pairs. Again, 23 from our mom, 23 from our dad. The mom and the dad chromosomes match up. Okay, chromosome 1, we have 2 of those, one from mom, one from dad. They match up, they have the same genes on them, but they are different versions of the gene. We'll get into that more as we do more activities with meiosis. This is your first introduction to homologous pairs, though. So if we review this pattern one more time, I want to make sure that you understand the process. In humans, we start with 46 total chromosomes, but 23, need to get my pen in play here, but 23 total pairs, okay, homologous pairs. For this diagram, I'm only showing you one homologous pair. In humans, when we go through meiosis, all 23 pairs have to go through this process together. So, all 23 pairs are going to partner up with their homologous pair. All 23 pairs are going to get copied. All 23 pairs are going to divide. For that first division, the pair split. Now we don't have homologous pairs anymore, right? They've split apart. At this point, we have sister chromatids. Hopefully remember that word from meiosis. So at the second division, the sister chromatids divide, and now we've got half the number of chromosomes, or half the number of DNA. So, second division, sister chromatids split. First division, homologous pairs split. In the end, we have half of our DNA in each of our egg or sperm cells. One more slide I'd like to show you is what this really looks like in actual cells. We're going to take a look at the testes tissue of mice and look at what their cells look like when they're going through meiosis to make sperm cells. So the first picture here is this parent cell. You can see the DNA just kind of looks fuzzy in there. You can't see individual chromosomes. And in the next picture, there's kind of like this dark blob. That's all the chromosomes lined up in the middle of the cell. This is where they'd be lined up with their homologous pair. Okay, so right here, the homologous pairs would be lined up. Next, we have our first division, where the homologous pairs are split. So the mom and the dad chromosomes are split apart. This does not mean that like all the mom chromosomes go this way and all the dad chromosomes go this way. You'll see this when we do an activity in class. But it does mean the homologous pairs are split from each other. Those chromosomes start to unwind and coil back into their separate spots on opposite sides of the cell. They line up again. Now they've got sister chromatids, remember? They're no longer in homologous pairs. This picture down here shows the sister chromatid splitting. So this blob up at the top is going to the left and the right. So sister chromatids are splitting. This blob of the chromosomes down at the bottom is going left to right also. So they're splitting the sister chromatids. And now we've got 4 distinct areas of DNA or chromosomes. Each of those gets a little membrane around them. And we've got what we call the 4 gametes. In this case, they're going to be 4 sperm cells because it's in the testes. So again, this is what it really looks like in real life, but you'll see the same pattern. First division, homologous pairs are split. Second division, sister chromatids are split. In the end, we end up with 4 cells or 4 gametes, and each has half of the genetic information in them.